Please start.
So welcome back everybody. Today is session 13 on machine learning and biology. First of all, I apologize to everybody here for being late. It completely slipped my mind that today's session was in the morning. So sorry guys. For today, our goal is to quickly first finish PCA, and then there are two very small topics remaining that also we will finish. And I will show you guys a very important, a very key thing, which will help you in analyzing transcriptomics dataset using machine learning, which is feature importance or SHAP. Okay. Once we have done that, then I will go ahead and explain to you what you guys need to do for the mini project. Okay. I will also share a document, a PDF of the instructions that are there for the mini project. Okay, and it'll all be clearly written there as well, but then I will also explain it to you once on call so that you guys can understand better. Okay, so with that said, let me share my screen and we will quickly finish the PCA. Okay, so today's lecture is going to be first with PCA. So PCA, as you all know, stands for principal components analysis. Okay, so PCA is primarily a dimensionality reduction technique. When I say dimensionality reduction technique, if you guys remember, what I mean is, say for example, today, if your features, right, if in your data set you have two features, or say even up to three features, right, you can still plot them, right? You can still plot three perpendicular axes on a paper or on a computer and visualize it, right? Up to three is still fine, but usually we have seen that usually we have way more than three features, right? And especially if you're going to be working with very high dimensional data such as that of RNA-seq, right, you would ideally have anywhere between 20,000 to 50,000 features, right? Because every gene, every non-coding RNA, every miRNA, etc., etc., each one of them is a feature, right? So when it comes to such high dimensional data, or in other words data that has a lot of features, right, it becomes very difficult for us to actually see clear patterns or visualize them, right? And that is where dimensionality reduction techniques like PCA come into the play. So what PCA particularly does is that it transforms correlated features into a set of uncorrelated components, capturing the maximum variance in the data. Okay, so just like we saw correlation heat maps, right? And I told you guys, if you remember, that if you see that any two features in your feature set is actually having very high correlation, right, you can go ahead and drop one of them, right? That is something that we end up doing sometimes, right? So PCA is kind of doing a very similar thing but a very different thing at the same time. Okay, so it figures out all the feature correlations and stuff, right? And then it transforms correlated features, so features which have very high correlation with each other, it transforms them into a set of uncorrelated components. So it creates new features, right? PCA takes the already created features, already the features which are already present, and creates new features which are known as principal components, which are nothing but a combination of correlated features. Okay, and the idea behind doing PCA is that each component, right, each principal component will try to capture the maximum variance in the data, or to tell you guys in easier words, it will try to uh, capture the maximum amount of information the data is trying to tell you in each principal component. This all will become a bit more apparent when we go into a little bit of the details of PCA. Okay, so our focus in today's lecture will solely be on why exactly we need to do PCA and how we can interpret results from a PCA plot. Okay, this is mainly because the math behind PCA is pretty complex, and it requires a very clear understanding of advanced algebra concepts like eigenvalues, eigenvectors, covariance matrices, etc. So we won't go into the mathematical know-how of PCA. It is absolutely not necessary for you guys. All we need to know is why we need it, and when we do it, how do we interpret the results. Okay. However, if you are interested in the math, you can check out this video. I have linked it here. This is a StatQuest video, and here Professor Starmer has explained the math in as simple words as possible. It is in no way, shape, or form the same math as these, right? But he has simplified it to the extent where even biologists can understand, who don't deal with math on a day-to-day basis can understand. So you can check it out. Okay, but it's not strictly necessary. But if you're interested, do check it out. Okay, so let us start. Consider this as our data set for the day. Okay, so these are our samples, obviously, people. This is our target column, which is lifespan. Okay, in years we are trying to predict how long a person will live, and these are all some particular metrics, right? So height is there, weight, blood pressure, heart rate, BMI, cholesterol, sugar levels, etc. So there are 200 features here. We have only shown 8, right? But there are a total of 200 features here. Let us assume. Okay. Now, obviously, we cannot visualize so many dimensions all at once, right? Obviously. I mean, this is exactly what we discussed. Now, is there a way of taking into account all the factors? So basically all the features. Is there a way I can reduce the number of dimensions, but then also take into account all the features, right? That is basically what principal component analysis does. Okay. So what PCA is going to do is, it is going to take all of these features and combine them in a very clever way, right? It will do a linear combination and produce new features like these, which are known as PC1, PC2, PC3, PC4, PC5, etc. Okay. So these new features are known as principal components. Okay. And it does this combination in such a way, right? So that if you focus on just the first few components, we will retain most of the information from the data set. So the idea behind PCA is, when you create new features from the original feature set, these new features are known as principal components. First thing about principal components is that each principal component is nothing but a linear combination of all the features here. Each principal component is a linear combination of all the features here. Okay. And as we go on, right, the principal components are ordered with respect to the amount of information they retain. Okay. For example, principal component 1 retains the most amount of information, so it explains the highest amount of variance in your data. Whenever I say something explains a lot of variance in your data, if you guys remember, that is what we did with linear regression as well. R squared, right, the R squared value was actually telling us how much of the variance in my data is being explained by the line that I have fitted to the data, right? So it basically tells me how much of the information has been captured by that line, right? And that is the same thing that we get here. How much of the information is captured by PC1, how much of the information is captured by PC2, how much of the information is captured by PC3, and so on and so forth. Okay. And the way the math it is designed is that PC1 will explain the highest variance, followed by PC2, followed by PC3, followed by PC4, and so on and so forth. And that is exactly why in when we do the differential gene expression analysis, when we plot the PCA plots, right, for the samples, we usually plot PC1 versus PC2, or PC1 versus PC3, and PC2 versus PC3, right? We do not go beyond PC3, because in those three features it has already it has already explained most of the variation that we need. Okay. Now there is still a problem here, obviously, right? That we still have 5 features here. Okay, say for example, we have done the principal component analysis and we have extracted the first 5 principal components. Okay, as a general rule of thumb, okay, and the way the math is designed here, you will get, so for example, there are 200 features, right? You will have 200 PCs as well. Okay, when you perform principal component analysis, basically what you end up doing is a lot of vector algebra. Okay, and if you guys are familiar with vector algebra usually leads to results which are of the same size of the uh, input or the query. Okay, so since there are 200 features, right, each of these PCs will be a linear combination of these 200 features, yes, but you will end up with 200 PCs as well. That is how the math works. You will get till PC 200. But the fact is that most of the variation or most of the information in your data will be captured within the first few PCs. So you don't have to use the PCs which are much later on, right? But it will be calculated and stored. Okay, but you don't have to really use it. So for the sake of this example, let us assume right now that we have extracted only the first five PCs. Okay, the first five principal components. Okay, now obviously we still have five features there, right? We still have five features, and we cannot really do a five axis plot, right? We cannot make a plot with five axes, and that is exactly why these rankings come in play. Right, as I told you guys, PC1 will have the highest ranking because it has the most information about our data set. Then we have PC2, then we have PC3, followed by PC4, and so on and so forth. Right? So in PCA, the principal components are ranked from most important to least important, always. Okay. So now let us go ahead and plot PC1 versus PC2. Okay, or in other words, these are going to be the two highest important PCs, right? These two principal components are the ones which are explaining the highest amount of variance, right? So say for example, this is the plot we end up with. On the x-axis we have PC1, on the y-axis we have PC2. Okay, lifespan here is being explained as a gradient, right? Towards blue is much older and towards yellow is much younger. Okay, so we can clearly see, right? So what are some inferences that I can draw from this plot, right? One very, very clear inference that we have is that our samples have clustered together really well. PCA, remember guys, please don't confuse this. PCA is not a clustering technique. Okay, the math and the algorithm behind PCA does not make it cluster samples. It makes it reduced dimensions. When we plot the principal components, we visualize the clusters. But the clustering is not being done by PCA. Okay, PCA is simply reducing the dimensions. Okay, the fact that these samples belong to older people is what is clustering them together, not PCA. Okay, I hope you guys understand that. Okay. So we can very clearly see that people who live longer, right? So these guys seem to be grouped together, and those who live for a much shorter time, right? They are also grouped together. So PCA technically in these, so we started out with 200 features, right? We started out with 200 features. Principal component analysis created two such features which is able to take into account all those 200 biological factors, convert them into new variables called principal components. And we have taken only the first two, and the first two is actually helping us get such interesting and such beautiful separation amongst our data, right? So this is something which is what PCA does. This wouldn't have been possible if you had 200 features, right? Visualization of your data, figuring out patterns of your data wouldn't have been a possibility if you had 200 features, right? But you can with principal component analysis. Okay, now obviously a very natural question now. We are using PC1 and PC2. What about the rest of the PCs? How do we know that the first two principal components are enough to capture most of the information or variance in the data set? Okay, a very valid question, right? That how do we know that only the first two is enough? How do we know we don't need to take care of the rest? We don't need to take into account the rest. A very, very valid question, right? The solution to that problem is by doing something known as a scree plot. Okay, the scree plot basically tells you how much variance of the data set, or in other words, in a much simpler words, how much information is being retained by each principal component. Okay, for example, in this case, you can see that PC1 explains 50% of the variation in your data set. Okay, so basically, this is the same thing as saying that in this particular data set, right, if you were to classify samples, right, or if you were to predict the lifespan using only PC1, you would have an R squared value of 0.5. This is what this is saying, that you would be, or in much simpler words, you'd be correct 50% of the time. So in regression, you cannot be exactly correct, right? But you get my point, you get the idea, right? That is what this guy is telling me, right? And let me tell you something in terms of real life data sets, right? Getting a PC1 which is greater than 40-45%, that is actually a blessing. Okay, in real life data sets, if you get anywhere close to 40% explained variance by the first principal component alone, that is absolutely amazing. Okay, we beg the data to do that, but most of the times it does not happen. Okay, so 50% is a good number. Obviously, the higher the better, but 50% is also a very good number. Okay, so basically, if you add PC2 to it, for example, right, you can see here it is somewhere around 35, right? So in this plot, basically, this axis, this axis is taking up 50% of the separation power, so to speak. This axis is taking up 85% of the separation power, so to speak, right? So these two axes together is basically taking up 85% of the separation power, right? So when I am using both PC1 and PC2 to do some prediction, I am able to do it 85% of the times correctly, so to speak, not exactly, because we are in a regression setting, right? So technically, I should say that my R squared value will be 0.85, right? But to give you guys the idea, that is what this means, right? And that is exactly why we can do away with only the first few PCs, right? PC3, if you see here, I think it is somewhere around 7 or 8, right? So if I add the three, right, if I use PC1, PC2, and PC3 as my features instead of all the 200 features, right, I am still kind of retaining 93% of the information that my dataset is giving me, and I don't have to use all the 200 features, right? So that is a win-win situation for me. I am reducing processing time and required compute for processing this dataset, and I'm still retaining most of the information that the dataset is giving me. Okay, so that is a win-win situation for me. Okay, and now of course, how much variance explanation you want for your dataset, that is completely subjective to you. It will change from study to study, but as a general rule of thumb, if both your PC1 and PC2 together, if your first two PCs are explaining 85% of the variance in a unit, in a quantity such as life expectancy, right, which itself is a highly varying data. If your first two PCs are doing so, if your first two PCs are actually showcasing an explanation of 85% variance, it sounds pretty nice to me. Why? Because life expectancy is itself such a high variance data, right? Life expectancy is such a high variance data anyway. On top of, even though it is so high, so much high variance, my principal components are able to capture 85% of that variance. That is good enough for me to do some sort of prediction. Okay, and visualization, so obviously I can definitely do visualization with a total explained variance of 85%. For prediction also, 85% is not bad. However, we can do better. we can use PC3 and PC4 to take it up to 95, 96, and we can do the prediction on that. So we can do better. Okay. So ideally, with most data sets, what we aim for is around 90% variance with just the 2 to 3 first PCs. Okay. Basically, the idea is that we need to retain enough information so that we can still visualize our data on a plot and do whatever classifications we need to do. Okay. Do people understand up to here what a principal component analysis does and how we kind of plot it? What is a scree plot? Right? Pretty simple concepts here. Okay. Okay. Now let us dive a bit more into the details and how we can kind of extract some more information from these plots. Okay. How we can extract some more inferences from these plots. So if I ask you what exactly is principal component one, what does it mean? It would be very difficult for you to answer, right? Right? You are a biologist who's interpreting biological data, right? You are interested in knowing which of the variables or features that you saw out of blood pressure, cholesterol, heart rate, etc., right? Which of those biological factors are actually responsible for the patterns we see among the observations or the people, right? You are interested not in, okay, you cannot say that in my experiment I saw PC1 was explaining 50% variance and it was able to separate this group from this group really well. What is PC1? There is no explanation for PC1. Nobody knows what PC1 is in your data, right? As a biologist who is communicating with other biologists, your target is to speak with respect to the biological factors or the features that were in your data set, right? So naturally, we would like to know which variables are influential and how the variables are correlated, right? If you guys remember, I said PC1, PC2, and every other principal component is nothing but a linear combination of all the features in your dataset, right? So it should be possible for me to figure out which feature is contributing how much to PC1, and that will tell me how influential it is and also how correlated each feature is. Okay, let us take a look here. So basically, whenever you perform PCA, right, you can also generate something known as a loading. Okay, these are known as PC loadings, right? Basically, what happens is each variable or feature gets a loading or weight for each principal component. This loading basically tells us how much that variable or feature contributes to that principal component. Okay, we can plot the loadings to see the relationship between all the 200 variables. Okay, we have only shown a few here, but we can do it for all the 200 and all the 200 PCs. Okay, so basically here for PC1, you see for height, the loading is -1. For average heart rate, the loading is 9. For BMI, the loading is 10, and so on and so forth. Okay, so loadings are nothing but a number that quantify how much this feature is important towards this principal component. Excuse me. Okay, that is what loading is all about. Okay, let me show you how you can actually interpret this a little bit better. This is known as the loading plot. Okay, this is known as the loading plot. On the x-axis, you have PC1, y-axis is PC2, and you simply plot this. Okay, say for example, we have plotted this for the PC1 loadings. Okay, let us focus on PC1 first. For PC1, which again is the most important principal component, right? Uh, we might expect to see variables like greasy diet, BMI, heart rate, right? This is heart rate, resting heart rate is here, etc. So these guys have really large weights, right? If you see here, BMI has a weight of 10, right? Heart rate has a weight of 9. BMI has a weight of 10, heart rate has a weight of 9, greasy diet has a weight of 10, and so on, right? Because why do you think this, why do you think that is? Because like, naturally, right, we said that these are weights, right? These are loadings, that means these are weights, and these basically tell you how much each of these features is contributing to this principal component. At the end of the day, this principal component is nothing but a linear combination of these features. So each feature is contributing something to the principal component, right? Now we can figure out the importance of each feature in each principal component by figuring out how much they are contributing, and that is what is given to us by these loadings or weights. Okay. So basically, variables like how many times you brush your teeth a day, for example, this has a much lower weight. Okay, if it is here somewhere, I don't see. You can see teeth brushing habits. Teeth brushing habits by default, with respect to the x-axis, is near zero. Okay, so this is something. So here you see it is zero point 0.2. Teeth brushing habits is zero point 0.2. So it has a much lower weight, right? So it is not as important. Of course, at the end of the day, we are trying to predict something like lifespan. So it would make sense that stuff like greasy diet, BMI, heart rate, frequent exercise, these are more important in predicting the lifespan of a person than how many times they brush their teeth. Okay, so that is very natural for us to see these results. So these are not results that just come up or just appear. They actually make sense, and that is what these loadings plot tell us. So now we have successfully converted PC1, right? We are not talking about in terms of PC1 anymore. We can actually trace back our steps and figure out what is giving PC1 the maximum contribution, which features are giving PC1 the highest contribution, right? And therefore we can make more informed decisions. If I say that I am differentiating two groups based on PC1, I am differentiating two groups based on the features which have the highest loadings on PC1. Okay, so that is something that I can kind of infer from this plot. Okay, now a very beautiful thing to notice here is that variables which contribute similar information are usually grouped together. Okay, variables which end up contributing similar information are grouped together on the loadings plot. Okay, so variables with a positive correlation are grouped together here, you see, right? If a variable is positively correlated with another variable, they are kind of conveying the same information, right? They are kind of conveying the same information, right? Because variable A is increasing, variable B is also increasing, right? For example, obesity and cholesterol levels, you might see that obesity and cholesterol levels in blood are positively correlated. That means if cholesterol levels in blood increase, right, obesity will also increase. If obesity increases, cholesterol levels will also increase, vice versa, basically, right? So the variables tend to change in the same direction. So to speak, the variables are conveying the same information, right? On the other hand, if cholesterol levels go down, obesity will also go down, right? So again, the variables tend to change in the same direction, right? If variable A goes up, variable B also goes up. If variable B goes up, variable A also goes up. If variable A comes down, B also comes down. If B comes down, A also comes down. That is the whole point, right? That is how positive correlation works, right? So two variables, they tend to change in the same direction, and that is exactly what we are seeing here, right? So variables that tend to change in the same direction, or in other words, variables which are contributing similar information, are grouped together. Do people understand this concept? This is a very cool inference that we can draw from PCA, and I'm sure you guys wouldn't have seen this before. So please tell me if you have understood this. Okay, only three of you. What about the rest? What about the rest of you guys? Okay. Now, naturally, variables with positive correlation are grouped together. So variables with negative correlation, such as height and resting heart rate, are in opposite sides of the origin. So these are not grouped together, rather they are on the opposite sides of the origin. Okay. So basically variables which are negatively or inversely correlated, right? When a variable is negatively or inversely correlated, what does that mean basically, right? That means if height increases, resting heart rate decreases. If resting heart rate increases, height decreases, something like that, right? So the variables tend to move in opposite directions. Increase in variable a causes a decrease in variable b, an increase in variable b causes a decrease in variable a, and vice versa. Okay, that is what inverse correlation or negative correlation is, right? If that is the case, right, these variables are positioned on the opposite sides of the plot, on opposite sides of the plot, or rather I should say on opposite sides of the origin of the plot. Okay, and they will always be in diagonally opposite quadrants. They will be in diagonally opposite quadrants if they are negatively correlated. Okay. So basically, what this is trying to tell me is that resting heart rate and height are inversely correlated, which is which is the same as which is the same as saying something like taller people tend to have a lower resting rate compared to shorter people, right? Because if your height is more, your resting heart rate is much less, is what this is trying to tell me, right? So variables which are positively correlated appear to be grouped together. Variables which are negatively correlated appear to be in diagonally opposite quadrants across the origin. Okay, that is the idea. And variables which have very high loadings, right? Which have very high loadings are the ones which are contributing the most, right? So in with respect to PC1 here, the ones which are to the extreme right or to the extreme left. Okay, that is the idea. Now, the distance to the origin also gives us information, obviously, right? So this is what I told you guys just now. The further away a variable is from the origin, right, the stronger the impact it has on my model. For example, here we can see stuff like obesity, blood pressure, average heart rate. They seem to be good variables to separate longer lifespans from shorter lifespans because they are way towards the left or way towards the right of the PC1 axis, right? So the distance to the origin also matters. A larger weight means a larger distance from the origin and therefore a much bigger impact. Okay, so this kind of a plot, which is known as a loading plot, right, it is a very good way of looking at all the 200 features at the same time and making some nice inferences and making some nice explanations about our data at once. Okay, this is kind of like a one stop solution. That is the whole point of this loading plot. Okay, so not only does it tell you which feature is more important, right, it also tells you the relationship between features, right, so to speak. So this is exactly how principal component analysis helps us identify this. Okay, so PC loadings, as I said, it helps us understand which variables are influential, how the variables are correlated. We started out with two targets, right? We started out with two goals to figure out which ones are more influential, which ones are less, how they are correlated. We have successfully understood how PC loadings plot can tell us both. Okay. Now the question is, right? Now we have one more question remains, that how do we use all of this information? Obviously, so we understood what kind of information it is trying to tell us. Now the question remains that how do we actually use this information to draw conclusions from our data set? Okay, let me explain with respect to this example. Okay, say for example, this is a gene expression matrix. Okay, and we have these expression values, we have these samples here, right, and we have this. Okay, so to get a general overview of our data, a good place to start is with PCA. Okay, so this is our PCA plot with respect to this data. Okay, this is our PCA plot. Now each point on the PCA plot is nothing but a single patient or a sample. Okay. And the idea is that obviously similar gene expression profiles will be clustered together. Okay. That is the idea. That similar gene expression profiles, patients which showcase a very similar trend in gene expression, will remain geographically in a similar area in our principal component analysis plot. Okay, because if you think about it, each PC is nothing but a linear combination of all these genes, right? So this is kind of a composition, or this is kind of a I should say, this is kind of an aggregate gene expression value. PC2 is also kind of an aggregate gene expression value, right? So we are trying to visualize these genes with respect to their expression patterns. And the idea is that patients or samples with similar expression patterns should remain in a similar region on the PC plot or principal component analysis plot. Okay. So just by looking at this plot, first of all, I can see that there are three clusters of patients, this, and this. Three very distinct clusters of patients. This means that overall, we have three very distinct gene expression profile. That is the first observation, right? And this is a very interesting one because it might mean that this group of patients will respond better to drug X, and this other group of patients will respond better to radiotherapy, right? For example, right, if these patients have different gene expression profiles, right, that now this will again be something which is specific to my study design, right? And this will actually tell me whether, for example, in this case, for example, these particular patients with such gene expression profiles, they are more likely to respond better to a drug that I have developed in my lab, which is X. These guys, on the other hand, with this gene expression profile, might be much better adapted to kind of go through radiotherapy and not the drug that I have prepared in my lab, right? So we are basically trying to figure out distinct gene expression profiles, and based on my study design, I can try and tell you guys that okay, this is for some treatment A, this is for some treatment B, and this is for some treatment C, right? And that way we can actually differentiate what works best for whom, right? That is kind of the idea. So now let us do a little bit more. Let's try and understand a little bit more. Let us focus only on the PC1 axis for now. Okay. The orange and the green clusters, right? The orange and the green clusters, they are very different based on PC1. Right? You can clearly see that they have a distance between them on the x-axis or the PC1 axis, right? So what this is basically telling you is that differences in gene expression profiles, right? Differences in the gene expression profiles of the green cluster and the orange cluster are likely due to genes that have very high influence or very high loadings or very high weight on PC1. So those genes which have very high loadings or very high weights on PC1 are actually contributing towards their separation on PC1. So those are the genes which are very differently expressed in these two distinct clusters, right? Remember, the loadings is what is telling us which genes have a heavier weight on the PC, right? So this way we can identify the genes responsible for differentiating between green cluster and orange cluster, as they are the ones with the largest absolute value of their loadings. Okay. So this is what this guy is telling us. The difference is mostly across PC1, right? So clearly the genes which are very important or with very high weights or loadings on PC1 are the genes which are differentially expressed between these two clusters, right? In case of pink and green, right, the difference is based on PC2. In with If I was looking at green and say for example, if PC2 was not here, right, the green would be somewhere here only. It would be overlapping with the pink. The green and the pink would be overlapping, right? Because with respect to PC1, they are at the same place. They are somewhere around here. They are localized somewhere around here with respect to PC1. So with respect to the genes which have very high loadings or very high influence on PC1, pink and green are not so different. But pink and green are different with respect to the genes which have very high loading on PC2, because that is where their difference lies. Their difference lies across PC2. Okay. Their difference lies across PC2. Now, another very important information we can get from here. Remember, the principal components are ranked. PC1 explains more variance than PC2, than PC3, than PC4, and so on and so forth. Right? So, the differences between clusters along PC1 are much larger than the differences along between clusters along PC2. Although they look very similar on the plot, and they will look very similar on the plot, right? We must remember that since PC1 is the one with the highest importance or the maximum explained variance, differences across PC1 are much larger than differences across PC2. So here clusters green and orange are more different from each other than clusters green and pink. Okay, the difference here is much larger than the difference here. Okay, so this is one more information that we can get from our PCA plot. A PCA plot is not a very simple tool that we simply do the plot and we figure out whether we are getting clusters or not. Okay, PCA plots tell us so much about our data, and it is very important for us to do PCA plots and visualize our data, to visualize trends and to visualize patterns in our data whenever we have extremely high dimensional data. So you can use PCA to visualize trends, jumps, clusters, and something that you have already covered during the RNA-seq part, that is to actually detect outliers in your data. Okay, so you can do all of this, and you can also use PCA to actually help do some feature engineering, which we'll see today. Okay, so do people understand this? Can you guys please give me a thumbs up if you understand principal component analysis and mostly how it works, how you can infer stuff from it? Yes, we did not go into the intricate mathematical details of it because frankly it's not necessary for you guys. You can always check it out. I have given the link to the video here on the PPT, so you can always check it out. Other than that, if you are still more interested in the math, I suggest learn vector algebra, learn linear algebra, and I mean, then the math will actually become much easier for you. Okay. Okay, I saw that only a couple of you gave me a thumbs up. What about the rest of you? Have you guys understood about PCA? Yes. There are 24 of you here. Come on. Okay. A few of you gave. Okay, no worries. Okay. Now, what we will do is we will actually do some scripting. Now, first, we will finish the scripting of PCA. So, what I want you guys to do is go ahead and download the PCA script that we have. Okay. So, we have this script, session 13 underscore PCA. Go ahead and download this, place it in the appropriate directory, and open it in PyCharm. Okay. Excuse me. Have you all downloaded the script and placed it in the appropriate folder? Can we begin? Please give me a thumbs up if we are ready to begin. Okay, I guess the ones who are following have done that. Okay, let me share my screen, and this is what we are going to be doing. Okay, we are going to be performing PCA on the same Wisconsin breast cancer dataset. Okay, now this again is the import section that we have. Okay, uh, so PCA is part of the decomposition library. Okay, PCA is part of the decomposition library, and there we have this function called PCA which will help us perform the principal component analysis. Okay, so here uh, I want to show you guys something. This is where obviously we do the same things, right? We load the data set, we drop the unnecessary columns, we encode target variables, we encode the target, etc., etc., all of that. Okay, and then finally we keep only the mean features, right? We keep only the mean features. That is what we have been doing in case of the Wisconsin breast cancer dataset. We don't really want to use the standard error or the worst ones. Okay, you can, obviously you can, but I mean, not really the most necessary. Okay, then obviously you will do standardization on these features, right? And then you will do whatever PCA stuff that we need. Okay, so let me actually go ahead and do that here. Okay, since we are doing a PCA here, uh, I guess let's not do this. Let us not do this where we select only certain features. Okay, instead we will basically use all the features which are present apart from diagnosis. Okay, and that is what we are doing here. Ignore this comment, this is wrong. Okay, ignore that comment. So for features, if you see here, we are taking all the columns, right, as long as the column is not equal to diagnosis. Okay, because diagnosis is our target column, right? So that is what we are going to be doing here. Okay, so here uh, this is nothing but our feature matrix, right? After that we'll do standard. Obviously again this is again during PCA, if you guys if you guys kind of notice, right, what is PCA? If you guys kind of notice, PCA also is very dependent on how each feature is, right? It creates PC loadings. It is a linear combination of each feature. So you want all the features to be of the same scale. You don't want a feature to dominate simply because its scale is higher than that of another feature. That is why it is very important for you to do standardization here as well. And that is what we're doing using standard scaler. Okay, so let me go ahead and put a breakpoint here. First, before I do that, let me uncomment all of these. Okay, you guys also go ahead and uncomment all of this. Okay, so this will help us actually do a little bit of the visualizations, right? Now, this is where we will get to do the PCA and stuff. Okay, so I will put the breakpoint directly here, because here all we are doing is we are defining the PCA function here. Okay, then we are doing the fitting and the transformation on the X scale to create the principal components. Okay, after that we are going to be calculating the explained variance, which can be calculated using the explained variance ratio underscore this function. Okay, it is part of the PCA function as well, which we are using here, right? And we are also going to be calculating something known as the cumulative variance. Okay, you'll see what these are. So let me put a breakpoint here and let me hit debug. So just give it a few seconds, it will run. So here you can see that PCA has happened, and it is the same, right? We have 30 features only. We have 30 features only, but now these are your principal components. So as I said, your 30 feature, you started with 30 features in the data set. So you will have 30 principal components as well. But it is just that the first few principal components are going to be the most important ones. Okay. So this is what I wanted to show you guys. Here, if you see the explained variance, this is the explained variance by each and every principal component. Okay, so this is in the scale 0 to 1. So obviously percentage explained variance is going to be 10 times of 100 times of this, basically. Okay, cumulative variance is nothing but you keep adding basically one on top of the other. Okay, you'll understand this. So let us quickly do the scree plot. Okay, you guys already know what the scree plot is. I'll quickly execute this and show you guys. Okay, yeah, this is giving me some trouble. Let me stop this execution, close this. Yeah, you guys might face some issues with this as it is a debug setting here. So that kind of happens sometimes. Let me try and close this and stop this somehow. Yeah. So yeah, that sometimes happens with matplotlib. Okay, you cannot really debug through the figure creation. Sometimes that happens. So what I'm going to do is I am going to put a breakpoint here after all these plots. Okay, so we will put a breakpoint at this point. Okay, we till the scatter plot of PC1 versus PC2, and we'll do the debug. Let it work. Yeah. So this is the first one, which is the scree plot here. Okay. So here you can see that in the scree plot, we are getting 0.44 as the explained variance here. So that is basically 44% explained variance by PC1, which is pretty decent. As I said, anything above 40 is pretty decent. Okay. For the second one here, you can see it is around 0.19. Okay. Rather, yeah, 0.19, right? So 19%. So the two together is doing 63%. Not too bad. Okay. So this is what the scree plot is telling us, how important each principal component is. Now this becomes a bit difficult for us to understand, that how many principal components should I retain? Should I retain 5? Should I retain 6? For my further downstream analysis, that becomes a bit difficult. That is why we do the second plot here, which is nothing but a cumulative explained variance plot. Okay, so if you see the code here for the cumulative explained variance plot, it is nothing but the cumulative variance, right? On the x-axis it is nothing but the cumulative, on the y-axis it is nothing but the cumulative variance explained, and on the x-axis the number of principal components which are getting used, right? So if you guys remember, the rule of thumb is to use as many PCs as it is required to get to at least around 90% explained variance, right? So if I actually see that around 90% explained variance, right, the next possible PC is this, which is 7. So using the first 7 principal components, I am able to explain 90% of the variance in my data, right, using the first 7 principal components. So I can actually use only 7 principal components. And when I'm using 7 principal components, the amount of variance I'm explaining is 91.35%, which is not bad, which is not bad at all. Okay. So we can use 7 principal components instead of all the 30 features. Okay. Now this is the scatter plot using PCA, right? So obviously the red ones are the benign one, are the malignant ones, and the blue ones are the benign ones. Okay, and you can see that the benign ones have clustered very well. The red ones are a bit more spread out. There are few outliers here and there. Obviously we can see few outliers are here, similarly here, right? So this is the plot for PC1 versus PC2. We already saw such a scatter plot and how these things are related, right? So we can see that the major difference is with respect to PC1, or the genes which formed a higher or the features rather which formed a higher loading or a higher weight with respect to PC1. Okay, now what we are going to be doing is we are going to be doing a 3D scatter plot. Okay, in order to do a 3D scatter plot we have to use plotly. Okay, we cannot use matplotlib anymore, we have to use plotly. Okay, so let us actually, in order to use plotly, you have to first create a data frame. So we are creating a data frame of the first three columns of XPCA, right? And then we are mapping it to the diagnosis column in the benign in the data set basically. Okay, so let me stop this. Let me remove the breakpoint from here and put a breakpoint here at 180. Okay, and I don't want to see all these plots again, so let me comment these out. Okay, let me comment this entire region so that we can save some time. And now let me hit debug. Just give it a couple of minutes. It is trying to render a 3D plot, so it might take some time. Yeah, yeah. So this is our 3D plot. Okay, so this has all the three axes. Okay, it has PC1, PC2, and PC3. Okay, here. Now, actually, the separation is a bit more apparent, right? If you look at it from this angle, actually, the separation is a bit more apparent. Okay, so this is where we are using all the three PCs. So all the three PCs would mean how much? That would mean around, I think, 70-75% explained variance, if I'm not wrong. Okay, this is what we are seeing. Obviously it is not possible for us to plot for anything more than three PCs together, so we will not do the plotting for any more, but here the differences are a bit more apparent. The From this angle we can see that the differences is much clearer. Okay, so sometimes three-dimensional plots help us in understanding the differences much better than two-dimensional plots, simply because you're adding more information to it. Okay, so this is what that is. I'll close this. We don't need these as well. And this is where we are. Now, let us actually see the loadings plot that we talked about. That is actually going to tell us which are the features which are the most important with respect to each of these species, right? Let us do the loadings plot for PC1 versus PC2. Okay, let us do the loadings plot. So for that, the first thing that we are doing is we are calculating the loadings. Okay, so the loadings comes from the components function in our PCA function. Okay, it comes from the components function. We have to first do a transpose of it. Okay, and then we can take the first two columns. Then only will we get the loadings. If I show you guys here, let me put a breakpoint somewhere here. Okay, let me put a breakpoint somewhere here and then hit debug. Okay, no, let me stop this. Uh, Let us actually comment out the 3D plot as well, otherwise it will take a lot of time. We don't want that. And now let me hit debug. Yeah, so this is our loadings plot. But before we go there, I want to show you guys this is what loadings is all about. Okay, let this load first. Okay, that will only happen, I think, after. Yeah. Okay, let me show you the plot then first. So this is our loadings plot. Okay, so this is our loadings plot, PC1 versus PC2. Here you can see a lot of these points clustered together with a very high weight, right? Similarly, here these guys are much closer to the origin than these guys, so these are not as influential. These are the ones which are more influential. And if you remember, guys, these are the ones that we also thought were more influential when we did our EDA, right? So we have perimeter, radius. These were the ones that were differentiating between the two classes the most, right? Now you also have these guys on PC2. You have this guy which is very high on PC2, tactile dimension. Okay. So obviously here we don't have a negative axis per se. Somewhere around here we don't really have one, because of the loadings. None of the loadings were as highly negatively correlated, right? That is why we don't see, but positive correlation we see here quite a lot. So these guys are very positively correlated with each other. So are these guys, so are these guys, and so on and so forth. So this is how you can do the loadings plot. Okay, and this is how exactly the loadings kind of look like. Okay, this is how the loadings look like for each feature, right? For the first two principal components. This is for each feature for the first two principal components. Okay, these are the loadings. These are the raw loadings that you can also kind of investigate. Okay, so now the idea is that we can actually build a model using the first few PCs. Okay, we can actually build a model using the first few PCs, right? So here we are using the first five PCs. Okay, the first five PCs is giving us how much? I don't remember, somewhere around 80 or 82% explained variance. So let us see how well our model does. And based on that, we can maybe increase or decrease. So now we have to be using our feature matrix, right? So our feature matrix is nothing but XPCA, right? And if you're going to be using only the first four PCs as our features, right? We have to use till 5, right? That is how Python works. Python works with -1 indexing, right? So here you see we have the first 5 PCs, not 4. Okay, let me change this typo. We have the first 5 PCs. Then again, it's train test split. You guys already know how this works. You do the split, you define the classifier, you define the parameter distribution, you define the parameter dictionary, right, for hyperparameter tuning. You set up the randomized search CV, right, and then you do the fit. You print the best parameters, the best cross validation accuracy, obviously, and then you will evaluate based on the best model. Okay, so let me actually stop this and run this as a whole. Okay, that will be better for us. Let me run this as a whole. We don't want this plot as well, so I can go ahead and comment this guy out as well. Now let me run this entire thing as a whole. After we have done the principal component analysis using the first 5 PCs as our features instead of the actual features. Okay, let me run this. Let's see what we get. So yeah, it is fitting. Don't worry. These are all warnings. Yeah. So you can see here that the best parameters were these. Okay. So these were the best parameters. We won't go into the we already know what these are. Okay, you're just saying np dot float 64 because these are all floating point numbers. Okay, that is the only thing. Okay, these are all floating point numbers, so these are the best parameters. One thing that I want to show you guys here is here I have defined the parameter dictionary kind of differently than to what we have seen earlier. Okay, here if you see, right, I have defined it not using discrete values, rather I am using these functions. So rand int is nothing but it will randomly choose integers between 100 and 500 to test for n estimators. Okay, it will randomly choose integers between 100 and 500 to choose for n estimators. Okay, so I earlier, if you guys remember, we did like 100, 200, 300, 400, 500 like that. Okay, here I am randomly choosing. Okay, it will choose such stuff randomly. Learning rate also, I am saying that choose anything randomly between a uniform distribution, from a uniform distribution, so that the probability of picking anything between zero point zero 0.01 and zero point 0.3 is the same. Okay, so that is why we are using a uniform distribution. That is why this uniform. Similarly, for max depth, we are using random integers. For subsampling, we are using a uniform distribution. All sample by tree, we are using a uniform distribution. Gamma, which is the gamma parameter, if you remember in XGBoost, right? That was used as the minimum loss required, right? For the pruning parameter, if you guys remember, we are using anything between 0 and zero point 0.5, but we are picking out from a uniform distribution because I want to make sure that the probability of every single value, of picking every single value, is the same. Okay, so that is the idea here, right? This is a bit of a different strategy. You can use discrete numbers here, as well as you can sample directly from these distributions. That is up to you. I just wanted to show you guys this. That is why I have it here. Okay. Now, the good thing to notice here is that using only the first five principal components, which was explaining around 80-85% of the variance in our data, right? The cross validation accuracy during training that we got is 0.978. So we got training accuracy, which is 0.978, which is pretty high. And similarly, we got testing accuracy also pretty high, which is 0.947. Okay, so we are getting almost a 95% accurate model with only the first five principal components. We did not need to use 30 features. We did not need to use 10 of the most important features, which are the mean. We did not need to use only 7 features out of all the means, which we had figured out using our EDA that these are the best ones. All we needed to do was do a PCA and use the first five principal components, and we are getting an accuracy of 95%. Right, so this is the power of PCA. Not only do you use PCA to visualize your data and see trends, you also use it to build models, to actually reduce the complexity of your models and build a model on these principal components to gain better accuracies. Okay, I hope people have understood till here. If you have, then please give me a thumbs up. Let us go group wise once. Okay, so if you have understood the theory as well as the code and the script of PCA, please raise your hand. Don't raise your hand, please give me a thumbs up. Okay, so group one. Okay, Akshaya, Samiksha, thank you. Pratima, thank you. Siddharth and Uday. Okay, group two. Varshini, Ananya, Vahaj and Nawaz. Okay, group three. Krishna, Sudiksha, Manaswini, Hasini, Lakshmi. Okay, group four. Hajra, Sayyeda, Sanjana, Nuseba, and Hadi. Okay. Group 5, Lasya, Harshita, Ramyashri, and Manaswin. Okay. Group 6, Haripriya, Tanmay, Sara, Soumya, Sai Krishna, and Sadhya. Okay. Group 7, we have Sahiti, Sadvedi, Pallavi, and Nikhil. Nobody from group 7 here today, is it? Sahiti, Sadvedi, Pallavi, Nikhil. Anyone? Okay. Group 8, Praneeth, Vidushi, Vedavati, Shruti, Shivani, and Sindhura. Okay, thank you. Okay, so we have understood PCA as well now. So with that, we can wrap up our chapter on unsupervised learning. So to give you guys a very quick recap, we learned about both supervised learning as well as unsupervised learning. Okay, now we have only a couple of topics left. Okay, and let us actually dive straight into them. Okay, so we already did PCA. Now we have something, this is, again, this is a very important topic. Okay. Uh, so we will learn about something known as an ROC AUC curve, something known as a PR AUC curve, and SHAP and feature importance. These are going to be the three other topics that we will learn. Okay, and so these are specifically, if you guys remember, these are nothing but evaluation metrics for a classification model. This is something we discussed way back when we discussed the basics of machine learning. And I told you guys that we will come to these once we have learned about the models. Okay, so we will do these quickly, and then we'll talk about something known as SHAP and feature importance. And this is going to be the key player in your projects as well. Okay, these three are going to be very key players in your projects as well. That is why I have kept them till the last. Okay, let us go into it. Okay, so the first thing that we are going to be learning about is the receiver operating characteristic curve, or the ROC curve. Okay, for that, obviously, we have to take a classification setting, right? So suppose this is the classification setting that we have, is obese versus is not obese, and we fit a logistic regression curve to the data. Logistic regression is used for classification, so we can fit a logistic curve to the data, right? So now this logistic regression tells us the probability that a mouse is obese based on its weight, right? Now, for example, if I get a data point here based on the weight, I will project it onto the logistic regression curve and say that the probability of this mouse being obese is around, say, 0.1, which is very low. Now, if the weight comes around here, I will project the data onto the logistic curve and say for this mouse, the probability of it being obese is around zero point 0.9. So logistic regression or any classification model predicts the probability. Okay, they are predicting the probability. Okay, very important to note here. They are not predicting whether the mouse is obese or not obese. They are predicting the probability of being obese, which is 0.1 in this case and zero point 0.9 in this case, right? Classes are assigned once we define a probability threshold. It is us as users when we are defining that I will keep a threshold of zero point 0.5, and that anything above zero point 0.5 will be classified as obese and anything below zero point 0.5 will be classified as not obese. That is where the classification is happening. So the classification remains in our hand, right? If we use probability of obesity as zero point 0.5 as the cutoff, right, we would call this mouse obese and this mouse to be not obese, right? That is what we would do, right? Because we as users have set the probability to zero point 0.5. Very important to note here. The probability is set by us. The model or the algorithm is simply predicting the probability value. Okay. So now what we can do is we can build a confusion matrix when we have set the probability to zero point 0.5, right? So once we have set the probability to zero point 0.5, right, we can do the testing of all the testing data, and on top of that we can build a probability, we can build a confusion matrix, right? So here we can see that these are the true positives, these are the true negatives, right, and so on and so forth. So this is a confusion matrix we have built, right, using zero point 0.5 as our threshold, right? Now once the confusion matrix is built, right, we can calculate sensitivity and specificity to evaluate how well zero point 0.5 did as a threshold, right? For example, now now a very natural question that you guys might have is that why is it even necessary for us to try out different thresholds, right? Why is it necessary? Why cannot everybody simply agree that zero point 0.5, which is the middle ground between 0 and 1, is the best possible threshold for classification, right? Why is that not the case? Because your data does not always conform to that rule, right? Suppose it was very, very important for us to correctly classify every obese sample, right? Suppose it is very important that I don't miss out any obese people. I have to classify all of them correctly, right? We could ideally set a threshold of zero point 0.1, right? In this case, what is happening? In this case, although we are misclassifying some people, right, but at least for all the obese people we are classifying them correctly, right? On the other hand, if we set a threshold to zero point 0.9, right, we are misclassifying some obese people, yes, but on the other end we are correctly classifying all the non-obese people, right? So suppose these come into the picture, right? For example, I mean, let me so this is with respect to obesity. Let me give you with respect to, say, for example, a cancer diagnosis, right? In a cancer diagnosis, it is extremely important for me to basically reduce the number of false positives. I don't want to trouble somebody by telling them that you have cancer based on this machine learning model, when they could very well be a false positive. Now they would have gone through a round of chemotherapy, and that would have given them some other disease that they were not even that they did not deserve, right? They did not even have cancer. I gave them the disease because my model diagnosed that this particular person has a cancer and they were a false positive. We don't really want that, right? So basically what happens is, when we change these thresholds, right, based on how we are changing the threshold, we can control how many false positives, false negatives, true positives and true negatives we are going to be allowing in our classification. Okay, for example, right, uh, we would correct when the threshold was set to zero point 0.5, right, this is what would happen, right? We would correctly classify the same number of samples, which are 3, right, and we would correctly classify one more sample which was not obese, which is 4, right? Now with this data, a higher threshold, right, which is a threshold of zero point 0.9, right, does a better job of classifying samples as obese or not obese. So this is with respect to a threshold of zero point 0.9. If I take you guys back to this is with a threshold of zero point 0.5. You can see here, right, 3 obese correctly classified, 3 not obese correctly classified, one obese incorrectly classified, one not obese incorrectly classified, one obese incorrectly classified. Now with zero point 0.9, we still have 3 obese correctly classified, but we have 4 not obese correctly classified. So we are actually doing better with a threshold of zero point 0.9 and not zero point 0.5, right? We are actually doing better. With this data, the higher threshold does a much better job of classifying samples as obese or not obese, right? Our higher threshold is actually doing a much better job. And that is why a globally agreed threshold of zero point 0.5 does not work for all of them, right? Because the threshold will be very specific to the data that you have at hand, the data you are using for training, right? So now the threshold can ideally be anything between 0 and 1. So the question is, how do we determine which threshold is the best? Right now, we decided that zero point 0.9 is a better threshold than zero point 0.5 because the confusion matrix told us so, right? So now, naturally, you can ask that, okay, do we actually build confusion matrices for every possible threshold, right? So if we actually do that, if we build confusion matrices for every possible threshold, it would result in a confusingly large number of confusion matrices, right? Look at the irony here. The very thing that is going to help us in determining which one is the best threshold is actually the one that is making it very difficult for us to determine, because there are so many to look at. I cannot look at 10, 15, 20 confusion matrices at the same time. And if you see between 0 and 1, every continuous value of a threshold can be tested, right? I can test anything between 0, I can test 0.12, I can test 0.59, I can test 0.43. So will I now go on building confusion matrices for each and every one of these thresholds? That is not humanly possible, right? That is absolutely not humanly possible. And this is the problem that this guy, receiver operating characteristic or ROC curve, is going to solve. Okay. uh, give me a thumbs up if you guys understand why it is necessary for us to test out different thresholds, and if you guys agree that other thresholds also work rather than zero point 0.5. right? Okay. Okay. Now give me a thumbs up if you understand why confusion matrices is such problematic in this scenario. Is it possible for you to look at 10, 15 confusion matrices at the same time and figure out which one is the best? No, right? I would say so. Only Pratima and Hadi are the ones responding. What about the rest of you? Are you guys even here? Or if you have not understood, please raise your hand. Please ask your question. I haven't had many questions. I think uh, I there were only two or three questions in the entire course of the internship. Okay, previously we used to have two or three or more questions in every class. Okay, so guys, please, if you do not understand, please speak up. I do see in the feedback form some of you are giving us feedback where you are saying that this thing went a little bit over my head. The math was a bit difficult. If that is so, then you have to speak up and you have to ask me the question. Then only will I be able to make the explanation even easier and help you understand, right? You have to do that during class. Okay, it is my request to you all. Okay. Okay, let us go ahead. Okay, so we have built the premise here, we have set the premise here that tells us that okay, we have to try out different thresholds because zero point 0.5 is not a universally agreed number one, and number two is we cannot try out different thresholds by actually plotting or actually figuring out the confusion matrices because that is going to be very cumbersome and humanly not possible. So we have to come up with a solution, the solution being a receiver operating characteristic curve or an ROC curve. Okay. So instead of being overwhelmed with these confusion matrices, we use this ROC curve to provide a simple way to summarize all of that information. Okay. So the ROC curve has two axes. The x-axis is known as the false positive rate, which is nothing but 1 minus specificity. And the y-axis is your true positive rate, which is nothing but your sensitivity. Okay. Now, obviously, we can calculate the true positive rate, which is nothing but sensitivity. True positives divided by the number of true positives plus false negatives, right? The true positive rate, basically, what does it tell us? The true positive rate tells us what proportion of the obese people or what proportion of the obese samples were correctly classified or what proportion of the positive class was correctly classified. That is why it's called a true positive rate, right? False positive rate or 1 minus specificity is given to us by the number of false positives divided by false positives plus true negatives, right? So it basically tells us what proportion of the not obese people were incorrectly classified, right? What proportion of the non-obese people were correctly classified, right? Not incorrectly classified, correctly classified, right? So it is telling us what portion of the sorry, incorrectly classified, correct, what portion of the not obese people were incorrectly classified, right? So how many false, so because we have false positives on the numerator, right? False positives are the people who are actually not obese, so they do not belong to the positive class, but our model has predicted they are belonging to the positive class or they are obese. So this is telling us the proportion of not obese samples that were incorrectly classified. So this is kind of a this is kind of a uh, this is kind of a metric to tell us how wrong our model is, and this is kind of a metric to tell us how right our model is, kind of. Okay, think about them in those terms, so this explanation will become much easier. Okay, now let us set a threshold here. Okay, let us set a threshold here, which is a threshold of 0, right? If we set a threshold of 0, right, obviously we will end up predicting all the people as obese, right, and nobody will be not obese. So we will have uh, 4 correct classifications as obese and 4 incorrect classifications as obese, which are actually not obese. Okay, so now we can calculate the true positive rate. The true positive rate is going to be what? 4 divided by 4 plus 0, which is 1. This means that every single obese sample was correctly classified rate was correctly classified. So a true positive rate of 1 means that I have made no mistake with respect to the positive class. Every single positive class sample or every single obese sample in our scenario was correctly classified. Okay. Similarly, I can also calculate the false positive rate. The false positive rate is also 4 divided by 4 plus 0, which is 1. This means that every single sample which is not obese was incorrectly classified, right? So my true positive rate is also the highest possible, which is 1. So in terms of being correct, I have done the best in terms of the positive, but in terms of the negative class, I have done the worst, right? So I also have a false positive rate of 1 and a true positive rate of 1. So every obese sample was correctly classified. On the other hand, every non-obese sample was incorrectly classified. We basically have both the best and the worst, right? So let us go ahead and plot that on this plot here. Right now we have a point. This is corresponding to threshold 0.0. You have a point at 1 comma 1. Okay. Now, a point at 1 comma 1 means that even though we correctly classified all the obese samples or all the positive class samples, we incorrectly classified all the negative class samples or all the not obese samples, right? So true positive rate is very high, which is very good. False positive rate is also very high, which is not good. We want a low false positive rate and a high true positive rate. We want something around this. Let us see actually how we can get to it. So if I now draw a green diagonal, right, from the origin to this point, this is the line at which PPR, true positive rate, is the same as FPR or false positive rate, right? Any point on this line means that the proportion of correctly classified obese samples is the same as the proportion of incorrectly classified non-obese samples. Any point that is on this line tells me that my model is so as good my model is with respect to classifying obese samples, it is as bad with respect to classifying non-obese samples, because this is the straight line on which every point is corresponding to TPR is equal to FPR. Okay, do people understand the green line? Can you guys give me a thumbs up if you understand the green line, or should I repeat? Okay, Hadi has given us a thumbs up. Pratima has also given us a thumbs up. Thank you, Lasya, Hasini. Okay, so people understand. Okay. So this green line is nothing but a baseline basically, right? At this point, nothing is changing. Your TPR, or how well you are doing with respect to the positive class, is the same as how bad you are doing with respect to the negative class. Okay, so this is kind of your baseline. Now let us try a new threshold. Okay, let us try a new threshold, which is say somewhere around zero point 0.1. Okay, this threshold is somewhere around 0.1, for example. This is the confusion matrix that we get when we use a new threshold. So instead of 4400 here, we get 4 here, which are for true positives. We get 1 true negative, and we have 3 false. We have 3 false positives basically, right? So now let us actually calculate the TPR and the FPR. So again, it will be what? 4 divided by 4 plus 0, and so 1. And this will be 3 divided by 3 plus 1, so 3/4 or 0.75, right? So we have the TPR and the FPR. Let us do a point here. Now we see that in this case, my false positive rate is still high, which is 1, but sorry, my true positive rate is still high, which is 1, but my false positive rate has come down a little bit. My false positive rate has come down from 1 to 0.75, right? Does that mean that this point is better than this point? What do you guys think? Is the newer point a better classification than the previous one? Yes, you think so, Hadi? Okay. What about the rest? Do you guys agree with Hadi that the new point is actually doing a better job at classification than the older one? Okay. And isn't that kind of evident from the confusion matrix as well? Earlier we had 0 here and 4 here. Now we are doing one more classification which is correct. and one less classification which is wrong. So obviously we have done something better, right? And that is what is being shown here. Okay. Now since the new point, now this is where we figured out that it is better with our eyes. Now actually look at it from the mathematical perspective, right? Since this new point is towards the left of the dotted green line, right? Anything to the left of the dotted green line will have a higher TPR than than an FPR. Anything which is towards the left, any point towards the left will have a higher TPR than FPR, and anything towards the right will have a higher FPR than TPR, right? So that is what we have, right? So this basically means that anything to the left has a higher proportion of samples which were correctly classified as obese, and a lower proportion of samples which were incorrectly classified as not obese, to be very technical with you guys. So we want points to lie to the left of our dotted line or baseline. Okay. So in other words, the new threshold for deciding if a sample is obese or not is better than the first one. We saw this with our eyes. Now we have also defined it using the graph. It has to be on the left. Okay. Now let us try another threshold. Okay. So for example, this new threshold gives us this confusion matrix. So we are making another correct prediction and two less wrong predictions, right? If I go ahead and calculate the TPR and FPR, I will see that we get a point here at 0.5 comma 1. So this point is the best out of all three of them till now. Okay. This point is even better. In other words, this threshold is the best one so far, because for this the TPR is the highest and the FPR is the lowest, right? That is what we have. Now the point at 0 comma 0, right? We will have, obviously we can build so many more confusion matrices and actually do these plots, right? uh, so basically what is happening is, this is what we will get: a point like a graph like this, or points like this. Okay. The point at 0 comma 0 represents a threshold, right, that results in 0 true positives and 0 false positives. Okay. Neither did we correctly classify any of the obese samples, nor did we incorrectly classify any of the non-obese samples. Or in other words, we incorrectly classified all the obese samples, but correctly classified all the non-obese samples. Okay, that is how you interpret. Okay. Now, the ROC graph kind of summarizes all the confusion matrices. Yes, at the end of the day, we have to build the confusion matrix, and then only can we get to this graph, right? Or rather, I should say, we have to calculate those true positive, false negative, true negative, right? That is what the confusion matrix is all about, right? So at the end of the day, what we have to do is we have to calculate those numbers, right? We have to calculate those entities, obviously, and then do the plotting on the graph. But when we are looking at it and when we are trying to make a decision, we don't really have to do it, right? We don't really have to look at all the confusion matrices together. Rather, what we can do is we can simply look at the graph and decide which threshold is going to be the best for us. Okay. So we can decide that. Okay. Now, without having to sort through all these confusion matrices, I can tell that this threshold is better than this threshold. Okay, because for both of them, the true positive rate is the same, but the false positive rate is less for this guy. So this guy is better than this guy. Okay. Now, depending on how many false positives I'm willing to accept, the optimal threshold is either this one or this one. Okay, so obviously you can argue that this one is better than this because this has a higher TPR, right? And now I can counter argue with you saying that no, this guy is better because this has the lowest FPR, right? So it all comes down to how many false positives you are willing to accept in your model. Okay, so for example, if you need your model to have a very low false positive rate, for example, an HIV diagnosis, right? If there is a false positive and I tell a person who actually does not have an HIV that they have HIV, right, it can cause severe psychological distress and unnecessary treatment, right? So the test must have an extremely low FPR, right? Even if some true cases are missed, I don't want to raise any false alarms. So in such a scenario where I need a very low FPR, I need to choose the threshold at which my FPR value is lower, which in this case would be this guy, right? If I am okay with getting a few false alarms, right? Because say for example in case of cancer, at least I am not missing out on any true values, right? I want very high, I want very low false positive rates is okay, but I want very high true positive rate, right? I don't want to miss out on anybody who has cancer, even though if that means that a couple of people have to be misclassified as having cancer. Okay, anyway these people will go and do the biomarker testing, right? So my model can kind of what should I say, can afford to be wrong in terms of the healthy people a little bit, as long as it makes sure that it does not leave out any of the cancer driven people, right? So in that case, a high FPR is acceptable to me, right? So in this case, this is acceptable to me because I am having a very high TPR, right? And a comparatively high FPR, which is acceptable to me. Okay, so the choice of whether this guy is going to be the best one or this guy is going to be based on how many false positives your model is willing to incorporate. Okay, that is how you are going to choose. Okay, both of them have their own pros and cons. Okay, that is how you are going to choose. Now this is how you choose the threshold using a receiver operating characteristic curve or an ROC curve. Okay, the area under such a curve is known as the AUC or the ROC AUC. So this red shaded region is the area under the curve, and this is 0.9. For this particular curve, the area under the curve is 0.9. Okay, now suppose we are trying out different models. Okay, suppose this red one is with respect to linear logistic regression, and this blue one is with respect to a decision tree. Okay, so we can clearly see that the area under the curve for logistic regression is much higher than the area under the curve for decision tree, which is blue, right? So this basically tells us that the red curve is much better. Okay, so if the red curve is something which is representing logistic regression and the blue curve is representing random forest, we would use logistic regression for our classification. Okay, and that is exactly why it helps us evaluate certain metrics, it helps us evaluate algorithms, right? So what this is basically telling me is that logistic regression as an algorithm is working much better than random forest across all possible thresholds, right? Logistic regression as an algorithm is working much better than random forest or decision tree across all possible thresholds. And that is why logistic regression is a better model. So the ROC curve tells you two things. The ROC curve by itself tells you which threshold to use for a particular algorithm, and the ROC AUC of different algorithms tells you which algorithm to use for classification. The one with the higher AUC is the one you're going to use, because that basically tells you that that particular algorithm is performing a much better classification overall across all these possible values of the threshold. Do you guys understand the importance of ROC AUC in determining a particular model's basically, what should I say, quality, right? So it is not only accuracy. So if there is one metric which is second to accuracy, which is very important alongside accuracy, it is the ROC AUC. Okay, these two are the most important, accuracy and ROC AUC. Please remember this. Okay, I hope you guys understand this. Yes, can you give me a thumbs up? I know Hadi, you are listening. So anybody else? Okay, very good. So this is what a receiver operating characteristic or an ROC curve is all about, and this is what the ROC AUC is, right? So although the ROC graphs are drawn using the true positive rate and the false positive rates to summarize confusion matrices, there are other metrics that attempt to do the same thing. Okay. For example, people can often replace the false positive rate with precision, right? People can replace it with precision. So precision is nothing but the proportion of the positive results that were correctly classified, right? So the proportion of the positive results that were correctly classified, that is what precision is, right? True positive divided by true positive plus false positive. So these were all classified as positive. Out of all the ones which were classified as positive, how many were actually correctly classified? That is what precision tells you, right? And obviously, whatever you guys did, right? You guys did for the ROC curve, you can also do for this precision, right? If you simply replace false positive rate with precision, you can do the same thing, right? Now, why would I even need to do that? Okay, the answer is pretty simple. If you think about it, right? If there were lots of samples that were not obese, right? A lot of these red samples which are not obese, in comparison to samples which are obese, right? Then your precision becomes a more useful metric than FPR. Okay, this is because if you see here, precision does not include the number of true negatives, right? It does not include the number of true negatives. Since it does not include the number of true negatives, it is not affected by a class imbalance, right? It is only with respect to the positives, right? It is only with respect to the positives, which are already less. That is why it does not affect you in terms of class imbalance, right? Because you are not even considering the other class in this calculation. So it does not really affect you in terms of class imbalance, right? In practice, this sort of class imbalance actually occurs when you are studying a rare disease, right? When you're studying a rare disease, you will have more of the negative class or more of control samples than of the actual disease sample, right? We already discussed this, right? That in case of a rare disease, this is a very plausible scenario, right? So in cases like these, where the positive class is much less in comparison to the negative class, you should use a metric that is not affected by the overwhelmingly large number of negative classes. So use precision instead of a false positive rate. Okay, therefore you will not be affected by class imbalances. In that case, the curve will become a precision recall curve or a precision. So this sensitivity is nothing but recall. Okay, it will become a PR curve or a precision recall curve, and the area under that curve will become PR AUC, and it is the absolute same thing as ROC AUC. Okay, it is the absolute same thing. PR AUC is nothing but ROC AUC for imbalanced classes. Okay, so I hope you guys understand this. Can you guys please give me a thumbs up if you understand ROC AUC and PR AUC? Yes. Okay. So now I'll take 5 more minutes and we will quickly finish uh, another very important topic. So remember guys, ROC AUC and PR AUC, in theory I will share the scripts with you as well for these. Okay, and we will see. Okay, so now let me show you guys two more concepts which are SHAP and feature importance. They're very small concepts, but these are going to be very important for you guys. Okay, so SHAP stands for Shapley additive explanations. So a very common problem with machine learning is that a lot of these models actually are not very explainable, in the sense we know that the model is classifying a certain sample as benign or classifying a certain sample as malignant and so on and so forth, but we don't exactly know why. What are these features which are actually helping us do this classification, right? And if you remember, that is exactly what decision tree tried to solve. Decision tree tried to make these models uh, explainable, right? Decision tree tried to make these models explainable. So it has been a problem that has plagued the world of machine learning for a very long time, right? That we need explainability. We need to know which features a particular model or a particular algorithm is giving more importance to, right? So which are those features which are driving the kind of what should I say, which are the features which are driving my particular prediction, right? And that becomes really important, because especially in terms of a gene expression data set, you want to know, right? You want to know what are these genes which are actually causing the differential expression pattern. What are the genes which are actually causing the differences between a disease condition and a healthy condition, right? So you need to know that. In order to know that, it is very important for you to quantify something known as feature importance. Okay, one way to do it is by using this thing called SHAP. Okay, so SHAP is basically a technique that is based on game theory and contributions, right? And it basically tells you how much each gene or each feature is contributing to your particular prediction and what that means. Okay, okay, let us understand this. So this is something known as a SHAP value plot or a SHAP feature importance plot. Okay, so let us actually see what this means. Okay, for example, let us take the example of MLC1. So MLC1 is a gene. Okay, now here you see this legend is that for feature value. Okay, red means high and blue means low. So this basically means the value of the feature actually in your data table. So in that data frame that you had loaded, right, what is the value of that feature? Is it a very high value or is it a very low value? Okay. In terms of gene expression, what would that mean? A high feature value or a high count value would mean high expression, and a low feature value or a low count value would mean low expression. Yes. Do people agree to this? Do people agree with this? Can you give me a thumbs up if you agree with this? Okay, Hadi, what about the rest? Okay, now pay very close attention here. For MNC1, we see that the reddish part is more towards the left and the bluish part is more towards the right? Now take a look at the x-axis here. This is the SHAP value or the impact on model output. Okay, we see that the red ones, so each of these, what you see, each dot or each point here is nothing but one sample in your data set. Okay, each dot here is nothing but one sample in your data set. So for MLC1, for each and every sample, right, it has basically colored the point based on this high or low feature value. Okay, and it has plotted it on the x-axis based on this SHAP value. Okay, so what is the SHAP value telling you? First, let us understand that negative SHAP value is associated more with a high feature value, or a negative SHAP value is associated with a high counts value or high gene expression value, right? So higher MLC1 expression is associated with a negative SHAP value. Okay, and a lower MLC1 expression is associated with a positive SHAP value, right? So this is basically telling us that a higher MLC1 expression is associated with the negative class. A lower MLC1 expression is associated with the positive class, right? If the negative class in my experiment is a control, then I would say MLC1 is lowly expressed in my controls as compared to my, sorry, MLC1 is highly expressed in my controls as compared to my condition, right? Say, for example, my condition here is some sort of a heart disease. So I would say MLC1 is lowly expressed in the positive class or in the people with heart disease as compared to the control. So MLC1 is a down-regulated gene in my particular condition as compared to the control. Remember, the negative side of the SHAP value is corresponding to the negative class or class 0, or in this case healthy controls. Okay, what this plot basically tells us is that reddish values or high feature values or a high gene expression value of MLC1 pushes the prediction of these samples towards a negative class or towards healthy controls, whereas low gene expression value of MLC1 pushes the prediction of these samples towards the positive class or the diseased class. Similarly, this gene CLDN9 is the opposite. You see blue or low values towards the left or towards the negative class. So low CLD9 expression is associated more with the negative class or the controls. and high CLD9 expression is associated more with the positive class or the diseased class. Okay, do people understand what this plot is trying to tell us? Please give me a thumbs up if you understand my explanation, my inference. And this is exactly what you have to do in your projects. Okay, you will be doing it based on gene expression data, and you have to figure out which are the genes which are the most important. Okay, anybody else would did you guys understand? Only Hadi gave us a thumbs up. What about the rest of you? Yes. Okay, very good. Okay, so this is what SHAP or Shapley additive explanations actually does. It figures out which are the genes which are the most important, and it tells you out of these genes how do they behave, how do they interact, right? Whether their high values is actually pushing the prediction more towards negative class or positive class, or whether low values are pushing it, and vice versa. That is what SHAP tells you, and you will be doing these SHAP plots to actually figure out your results. Okay, another way of doing this is by using something known as feature importance. So feature importance is not something that is available for all kinds of machine learning models. Okay, it is mostly available for those machine learning models where weights are directly used. So in case of logistic regression, SVM and SVM, feature importance is very easily available. Even in case of random forest and decision trees also, you can get the feature importance. Okay, but for boosting methods like gradient boost, XG boost, all of them, you have to use something like SHAP or Shapley explained variables. Okay, so here again, this is a very similar plot as to the other one, but this is based on feature importance. Okay, so on the x-axis, instead of the sharp value, you have something known as the coefficient value. Okay, and on the y-axis, you have the names of the genes. Okay, so here we are assuming obviously that 0 is our control or negative class, and 1 is our condition or positive class. Therefore, a negative coefficient indicates down regulation of said gene in my condition as compared to control, and vice versa. Say for example, SRSF7. SRSF7 has a negative coefficient value of minus 1 point something. That means this gene is down regulated in my condition with respect to my control, and the magnitude of the coefficient indicates its importance towards prediction. So this is basically telling me that in terms of predicting the control class, in terms of predicting the negative class, SRSF7 is really important, because this is what is driving the prediction towards the control class or the negative class. Similarly, for this guy, L ln RF1, this guy is driving the prediction more towards the positive class or the disease class. So this guy, which has a higher coefficient, is upregulated in my condition with respect to my control. This guy, which has a negative coefficient, is downregulated in my condition with respect to control and pushing the prediction more towards the negative class. Okay, this guy, which is upregulated, is pushing more towards the positive class or the disease class. And the magnitude basically tells us how important they are for the prediction, right? How influential they are for our prediction, right? So this is another way of looking at feature importances, which is using raw feature importance in case of models like logistic regression, SVM, and random forest and decision trees. Okay, for gradient boosting methods, you have to use SHAP. You cannot really use feature importance because they don't exactly work with weights. Okay, so this is something that you guys will do. I have a script here that will help you perform ROC AUC as well as SHAP as well as feature importance. Okay, uh, this is not too much of a difficult script. The only thing that I want to show you guys is that you have to install another package for SHAP. So all you have to do is go to your terminal, right, and go ahead and type pip install SHAP. Okay, and then it will install this particular library known as SHAP, and you'll be able to use this script. Okay, and for the ROC AUC as well, from the metrics module, you can import these ROC curve and ROC AUC score, and you can do the plotting here. I have shown here, I have shown it in the code, and it is basically very well explained. So what you guys will do now, this is your homework. What you will do is you will take this script and you will execute it on your own. Okay, you will try to execute it on your own and understand. If you have any doubts, you will get back to me. We anyway have one more lecture before your finals, before you finally present your results to me of your projects. So we can discuss there, as well as you have Telegram to actually discuss this with me. Okay. So I want you guys to actually run this script on your own, execute it on your own. If there are any errors, show it to me on Telegram. I will help you fix it. Otherwise, try and understand it. If you have any gap in understanding this, then also you can let me know in Telegram or in the next class, and I will take it up with you. Okay. But I want you guys to work on this now. Okay. Today is our last lecture. After this, you have to start working on your own. Okay. So you have to do this on your own. Okay. I will not run this for you and show this for you. You guys can do it on your own and come to me with questions. Okay. This also uses the same data set, which is the Wisconsin breast cancer data set. Okay, so you guys can get an idea. So it should not be too difficult for you guys. Okay, so be very well informed with respect to that. Now, with that, we have come to the end of our lectures and our course material, so to speak, with respect to machine learning and biology. I have taught you almost most of it. Yes, and this is what you will need for your future analysis, whatever you need. Obviously, you can always try and learn a bit more. You can always explore on your own, and you should. I feel that you should. But as far as the scope of this internship is concerned, we have reached the end. Okay. So if you have any questions with respect to anything that we have learned in the past week or so, please raise your hand. If you do not have any questions right now, it is absolutely fine. Please review the lectures, please review the recordings, and please try to understand. If you don't understand, come with questions in Telegram. I will help you answer them. Okay. Please do not feel shy, or do not, spend some time with this so that you guys internalize, so you guys don't forget this 1 week after the end of the internship. Okay. Please spend some time on this. Okay. It is my earnest request to you all. Okay. Now, with that said, we are done for the day. I will now explain the mini project to you all. Okay. So please pay very close attention. You will also have this document with you. I will share it immediately after the class. Okay. But please pay attention to this as I am saying. Okay. So on this mini project, you will have two parts. Okay. Part A and part B. Part A is going to be an exploratory data analysis. And this is something that each and every one of you will do individually. This part is not a group project part. Okay. You will do all of these individually. Okay. For part A, it will be generic and it will be the same for all of you. You guys will be working on the same data set, right? And its purpose is basically to make everyone practice EDA and pre-processing. Okay. So the goal for part A is that you will have this data set, which is known as the IRIS data set. I have given the link to this data set here. I will also upload it on the Google Drive. Okay. So you can always go to this link and check out more details about this data set whenever you want, right? What I want you to do is you have to perform EDA, right? You have to perform exploratory data analysis, right? And then you have to submit to me one .py file, which is going to be the code you used to perform the EDA, and one document or a PDF, whichever you prefer, on the results you got and the inferences you draw from your EDA. Okay. So this document or PDF, or it could be a PPT, whichever you prefer, I don't mind, whichever you prefer. This should be number one this should be filled with the plots that you have generated during EDA. For each and every plot, you should write down what you have observed, and you should write down what inferences you can draw from those observations. Okay, if you want, you can also do exploratory data analysis, like you can also give the outputs of the other EDA metrics, like for example summary statistics and etc. If you want, this is not something that I have taught you guys, but I have told you guys that a lot of people also perform statistical tests. If you want, you can also try and perform statistical tests. If not, that is absolutely fine. Okay, I don't expect you to perform statistical tests, but I do expect you guys to do the plots that I have shown you and draw inferences from them in the way that I have shown you. I will not repeat how we have done that. You can always go ahead and revisit those lectures and actually figure out what I exactly want from you. Okay, I have given you a hint right now what I exactly want from you. You will be graded on this. Okay, this will be a part of your grade. And as a matter of fact, for all of you, let me tell you this, this will be a part of your group grade as well. Okay, so if in a particular group a certain person uh, fails to submit this, or a certain person does a very bad job of this, right, it will affect their overall group grade. Okay, so if you don't want your overall group grade to get affected, make sure your group members are working hard on the EDA part as well. Okay, so this is to be done individually. The last date for submitting this is 23rd January 2026, 5 p.m. I will strictly not accept any submissions after 5 p.m. If it goes to 5:01, you will not be allowed to submit and you will be getting a 0 on this part. I am very strict about this. This is your EDA part of the project. Okay, this has to be done individually by every single one of them. Are you guys clear on part A? Can you please give me a thumbs up if you guys are clear on part A? Okay. Now for part B, what will happen is, this is where you will work with actual gene expression data and you will build some models. Okay. So each group will be given a single CSV file. The file will contain 19,299 columns. So I have already done the RNA-seq part of it, and I have prepared the data set and made it ML ready for you guys. Okay, so that you guys don't spend too much time doing that, since we have a very short time span. I have already done that, and you will receive a single CSV file with 19,299 columns. Is anybody trying to say something?
It's myself, Suryadev. If
Yeah.
possible, could you please maximize the screen?
Uh, how do I maximize this?
Because the font size is bit low. It should be. If or you can maximize the font size.
Is this better?
You can minimize some. Yeah, it's fine now.
This is better, right?
Better. Yeah.
Yeah.
It's visible.
So for part B, guys, each and every group will receive a CSV file with 19,299 columns, right? Out of these 19,299 columns, 19,297 will be corresponding to genes, and there will be two other columns, one being label and the other being batch. Okay, this is I wanted to make this as similar as possible to a gene expression dataset for a particular condition, so that is why you will have the label and the batch column as well. Okay, now one thing, fair warning that I want to give you guys, this is not these are not datasets that are coming from real RNA-seq experiments. It is not humanly possible for us to collate and correlate 200 300 samples from an RNA-seq experiment like that. So what I have done is I have created this data for you. This data is synthetically generated, but it is generated very carefully in order to make sure that it grabs the underlying biology of these conditions. Okay, so you can treat it normally, but do not try and perform any EDA on this. I mean, in general, it is not a good idea to perform the EDA that I showed you guys on gene expression datasets, because gene expression datasets do not really conform to that kind of statistical measures. Okay, so you won't get any decent, what should I say, decent results with respect to any EDA you do. on these data sets. So I would suggest do not waste time doing that. Go straight towards building your model. Pre-processing, yes, that is something that you have to do. You have to do the pre-processing, but EDA I don't think is necessary. Okay, so also this is not something that you will be able to open in Excel. So you would have to directly load it into a pandas data frame and deal with these columns. Okay, now this is step by step what you have to do on each of these CSV files. So on this CSV file, you have to first load it into the data frame. You have to define the feature and the target matrix. You have to perform any form of label encoding if required. You have to perform feature scaling if you feel it is required. Then you have to train and test classification models. Okay, you can train and test as many models as you like. Report the results of only 3 in your presentation. When you are making the presentation and when you are putting the results in your presentation, you should report only a maximum of 3 models, not more than that. I don't care if you have built 8 or 9 models, you can, but report only 3 models in your presentation. Okay, you can choose whichever 3 you want. If you want to report the best 3, it is up to you. If you want to report the worst somewhere as the worst model, the best model, and somewhere and one which performs somewhere in the middle, that is also fine. So that I leave up to you. It is up to you. Okay, but a maximum of 3 models. Okay, uh, there is no such thing as a minimum, right? If you only report one model, that is also fine, but you will be graded accordingly, because if you are only reporting one model, I will assume you have only done one model. Okay, so I am telling you guys all of this in all fairness, so be prepared for that. Okay, now once you have reported the results of the classification, right, you already know what to report and how to report. Make sure you guys do cross validation, make sure you guys do accuracy, PRAUC, all of that, right? uh, Apart from that, after you have done evaluating the models and figuring out, okay, this is the best model, now what you have to do is you have to identify the top important features using either feature importance or SHAP to infer the results of your model. Now this ideally you can do only on the best model that you have. Okay, if you want to do it on all 3, that is up to you. Again, I leave that up to you. You can showcase the results of all 3 as well. Again, that is up to you, but you can also do it only on one of them, right, which is ideally the best one, the best performing one. Okay, so feature importance using either the feature importance plot that I showed you guys or the SHAP plot that I showed you guys. Okay, and there should be inferences. Your PPT should strictly contain accuracy, precision, and F1 score at the very least. Okay, so these are the minimum requirements. Okay, so it should contain at least the accuracy, precision, and F1 score, the ROC AUC, the confusion matrix, and the SHAP summary plot or the feature importance plot based on the model you are using. Okay, after you have done that, obviously, as I have been saying, um, your analysis your analysis is only as good as the biological interpretation of the features that you have. Okay, so what I want you guys to do is, after you are done doing the SHAP plot, you would have identified the genes which are the most important for your classification, right? Now what I want you guys to do is search for those genes on the internet, look for literature with relation to those genes on the internet, and the condition versus control classification that you have done. Okay, try to relate the function of those genes with respect to the condition that you have studied. I want biological I don't just want to see that this is my model, this much accuracy I got, and these are the genes which are most important. I also want to see how these genes that are most important is actually tied to the biology of the disease you are studying. Okay, so that should also be part of your PPT. Okay, so you will be graded on the quality of your PPT as well, and also in terms of the content of the PPT. Okay, so whatever I have mentioned here is the bare minimum that you should have in your PPT. Anything you do more here, and if that actually makes sense and is correct, that will gain you extra points. Okay, you can be so these are the places, right, where you are doing classification, right, where you're reporting the results. This is where you can get a bit creative. You already know. I have showed you everything that you need to know in order to build good models. I have showed you everything that you need to know in order to figure out overfitting, underfitting, and all of that. So please review the lectures and do this exercise very earnestly. Okay, you will be graded on this. And obviously, as I said, these are the bare minimums. If you do something extra on top of this, it is only for you to gain. Okay, so do that. And obviously part A has to submit it a day before, which is 23rd January, so that I can finish grading them before we start our session on 25th or on 24th. Okay, so please do that. And as I said already, you guys will pay for others if your group fails to submit this on time, or if your group members are not doing this earnestly. The entire group score will go down. Okay, so please make sure that your group members are working with you on this, and make sure that your group members are responsible and you guys do this earnestly. Okay, do people understand the objectives of the project? Can you please give me a thumbs up if you understand the objectives of the project? Does anybody have any questions with respect to the project and how you're going to do it? You can raise your hand or put them in the chat. No questions? Okay, no worries. We have one more session before the final session where we can discuss any and all related questions to the project. Also, you can use Telegram to reach out to me for any doubts you have on the lectures as well as the projects. Okay, I will upload all the data sets here under data. Okay, so you will have both the iris as well as group wise data sets. Okay, I will assign each group one disease area, and I will assign the projects accordingly. Okay, and so each of these files will be named with the name of the group, followed by the disease area, followed by .csv. Okay, so only work on the data set that has been assigned to your group. Okay, do not work on the others. Okay, I hope that is clear to everyone. To clarify, EDA has to be submitted in the form of a Python script as well as results, right? Yes. So EDA has to be submitted as both the Python script and the PDF or the doc. So we will create a Google Classroom assignment for part one. For part A, and you can directly upload it there. Okay, for part B, you don't need to provide the Python script. You just need to provide the PPT. That also Bhavani will share a drive link for you to upload the PPT onto the folder. Okay, you have to upload it a few hours before our actual class. Okay, and then that is the PPT that you're going to use when you're going to be presenting. Okay, that will happen just like you guys did in phase one. I think you guys have already done that in phase one, right, Bhavani?
Yes, Surit. Whatever they followed for phase one will be the same for phase
Will be the same for phase two, but only for part B. Part A, you will create an assignment on Google Classroom and you will upload the Python script and the PDF or the document there. Okay. Okay, with that said, I guess I am done from my end. If anybody has any more questions, you guys can ask. Otherwise, we can wrap up for the day, and all the best to you all for your projects.
And Surit, before concluding, we need to think. Yeah. Dear students, can you please say us when your regular session and regular college is going to start? And if possible, we also need from tomorrow. Okay. College timing, please. Is that possible for you to go home before 5 p.m. because our session starts at 5, 9 to 4. And so I guess it will be a bit hard for them to reach home before 5.
Okay. So what do we postpone that session a little bit?
Whether you're available, next session is on 21st. Obviously, attending at 5 p.m. is not possible for them.
So it's okay. We can postpone a little bit. I am fine.
Let me know your available time.
Yeah, but we can do so. 6 p.m. would be okay, right, guys?
6
Can you give me a thumbs up?
The doubt clarification won't take. So,
time.
do you have any content to be clarified on that day apart from doubts by the students?
No content to be
Right,
clarified.
then it won't take more than an hour.
No, absolutely. So we can even so 20,
6:30 will be fine.
yeah, even 6:30 is fine. Let me just check. So we have it on
You
21st,
kindly
right?
check with us any time between 6 to 7 we can start.
21st is a Wednesday. Yeah, we can start 6:30 or 7. That is fine with me.
Okay. It's now students, please answer us whether a 6:30 session would be fine for everyone here.
Guys, please give us a thumbs up if 6:30 is okay with you.
Okay. It won't take much time, guys. So
Anyway, it's a doubt clearing session. So
Mhm.
if I mean, it is absolutely optional. If you don't have a doubt, you don't have to come to the session. Okay. I would suggest you come to the session because others' doubts will actually help you, but you can choose not to. I mean, anyway, a lot of you are skipping these sessions, so yeah, it's fine, I guess. Okay, so 6:30 is fine.
Yes,
Uh,
and 24th Saturday is our
huh.
final
So
assessment, right?
24th also, I think then let us start a bit late
I don't think so.
because
They'll be having work. It's working day for them on 24th since it's a Saturday.
guys, can you please confirm is Saturday a working day for you?
Or if possible, shall we make it on 25th if you are available, Suryadev? I guess they need at least a
Huh.
daytime. 8.
Saturday is not a working day for them. I think that Pratima, it's not a working day, right? It's holiday.
It's holiday.
So then we can do it on Saturday only.
Saturday itself, shall we have our assessment then?
Okay. Yeah, I am fine with Saturday. 20, Sunday would be 25th.
Yeah,
I
yeah.
am fine with Sunday as well.
Sunday. Okay. And now it's in the hands of students. Do you want it on 24th or 25th?
Sir, could you do it on Sunday, please?
I am
If
okay with
you do it
Sunday.
on Sunday, we should work
I
harder.
I am
We want
absolutely
to
okay with
balance.
Sunday. I leave the decision up to you guys. So is Sunday like a unanimous decision? Can we get a thumbs up for Sunday?
If it is on Sunday, timing will be same. It starts around 5.
Okay, so we have a unanimous decision for the assessment on Sunday. And so then let's have it on Sunday 5 p.m. No problem.
I think
No worries.
we can also reschedule the submission of individual assessment.
Yes. So in that case, guys, the the part A has to be submitted by 24th January. Okay. But 5 p.m. Okay.
Yeah,
No extensions beyond 5 p.m. Okay. Absolutely no extensions. I'm already giving you 24 days extra. No extensions on this. Okay. 24 hours extra. Sorry. Yeah.
all right. Then I think everything is fixed as of now. One small clarification. Your individual assessment has to be submitted on 24th and group assessment is scheduled on 25th. Okay. I think this is clear. Yeah. Also, I think they can submit their assessment in classroom itself, or do you
Yes.
want to share them a drive, which will be convenient for you?
No, drive is not required. Classroom, if you create an
Classroom.
assignment, it will automatically create a drive folder for me.
All
So
right.
just create an assignment. I will check it from there, just like last time.
Fine. Yeah.
Oh,
So, dear students, we will be creating an assignment in Google Classroom where you will be submitting your individual reports on 24th before 5 p.m. And group assessment is on 25th, and you will be submitting your PPTs before 3 p.m. to us. Yes. And as we followed for phase one, the assessment timing is for 15 minutes, 12 minutes for presentation, 3 minutes for discussion. Only one representative from each group will be allowed to present. And on a final note, please convince all your friends. We need everyone to take part in the group assessment. If we miss anyone, you won't be getting your certificate. With this, let's wind up for today then. Thank you, Suryadev. Thanks for your time. And thank you, students. Please do fill the feedback form. Meet you all on 21st, 6:30 p.m. Thank you.
thank you, everyone. I'll see you again on 21st. Thank you, and I will upload the datasets now.